So in this unit, we've been talking about the central dogma, which is when DNA is converted into mRNA through the process of transcription, and using the genetic code, mRNA is converted into protein in the process of translation. Last class, we went into a bit of detail looking at the process of transcription, and we did this looking at bacterial transcription. We then went on to look at an example of how transcription is regulated in bacteria using the example of the lac operon. In today's videos, we're going to look at how eukaryotic cells finish transcription. So there are a few extra steps to processing the RNA during transcription in eukaryotic cells. And then we'll look at how eukaryotic cells control gene expression. And this process is a little bit more complicated in eukaryotic cells compared to bacterial cells. So that's what we'll be talking about. Okay, let's review bacterial transcription. So here, this rectangle represents a gene in bacteria. And every gene has a promoter. And recall that the promoter has -10 and -35 box. And the promoter is where the RNA polymerase binds. And this RNA polymerase we call the holoenzyme because it's made up of RNA polymerase and another protein called sigma. And sigma is what helps the RNA polymerase recognize the promoter. And when this binds, the RNA polymerase opens up this DNA double helix. That's what this rectangle is representing. And it can transcribe a gene, right, into RNA. Now, in prokaryotes, when the transcription of mRNA is complete, the mRNA is ready to be converted into a protein. And of course, this all happens in the cytoplasm because there's no nucleus. This process can be slightly more complicated as I'm depicting above because many genes are regulated, like the lac operon. And we'll finish talking about the lac operon in lecture, but just to remind you, there are several genes that are regulated by one promoter. And there's an area that can serve in the DNA that can serve as an activator, that can enhance gene expression, and an operator that can recruit proteins to dampen gene expression of these genes. So this process can be regulated, but all of this is slightly more complicated in eukaryotes. So the first thing I'll talk about is that in eukaryotes, mRNA is not initially ready, and we call it pre-RNA. And this is the first thing that's produced in transcription, and it must be processed before translation can occur. So let's just review really quickly some of the things we've talked about that are different in eukaryotic transcription. So the promoter regions are both longer and more diverse. Many more proteins can bind to the promoters, and they're recognized by general transcription factors. Another difference is that this poly-A, there's a signal towards the end of the gene that's a poly-A signal, and this triggers a poly-A termination and then a poly-A tail to be added. So at this stage, the mRNA is not mature once it's produced. And as I mentioned, we call this the pre-mRNA or the primary transcript. So we have transcription, we have our mRNA, but this isn't ready yet. It's a pre-mRNA. And it needs to be processed before it can leave the nucleus and be translated. So in the nucleus, the mRNA becomes mature because of mRNA splicing, the addition of a 5' cap, and the addition of a poly-A tail. And I'm going to describe the mechanisms by which mRNA is modified in these ways to become mature. So first, let's talk about this RNA splicing. So to do this, I want to introduce the concept of introns. So there was a great experiment done early on where they took viral DNA and they heated it so that the hydrogen bonds between the base pairs separated like they would in a PCR reaction, and this produced single-stranded DNA. The scientists then incubated this single-stranded DNA with mRNA. And they thought that they would polymerize 1-to-1, right? Because in the process of transcription, DNA is just being copied into RNA. And what they saw was that loops formed. Okay, so that's shown here in this micrograph. And so they saw that these loops were forming when you incubated single-stranded DNA with RNA. And so here's a loop, and here's a loop, and here's a loop. And so what they interpreted was that there were stretches of DNA not present in mature mRNA. So in this diagram here, the mRNA is shown in yellow or in orange, and the DNA is shown in red. And you can see that as this is polymerizing, there's a point here in which this part that's forming complementary base pairing with the DNA, right, it polymerizes to a separate part of the DNA. And this is what forms these loops. And then here, right, this doesn't polymerize to this part of DNA, and so it's excluded in a loop. And so another way to think about this is that a 1-to-1 ratio of DNA to mRNA did not exist. So what's going on? So it turns out that many genes have these regions of DNA called introns. And these introns must be removed from eukaryotic RNA transcripts. So an intron is a stretch of non-coding DNA not present in the final mRNA, okay? And a way to remember introns is in the trash, okay? So we're gonna throw these away. The other parts of the gene that end up in the primary transcripts are called exons because they are expressed in the final mRNA. So hopefully those can help you keep straight what's an exon, meaning expressed, and an intron in the trash. So these are all made into mRNA, but then in the final product, they are spliced out. So how does this happen? So we're going to describe the process by which the cell splices the mRNA into a mature transcript. So the first thing that happens is that these complexes called SNRPs, which I think is an adorable name, bind to the intron region of a gene. Okay, so SNRPs are small ribonuclear proteins, okay, that also contain nuclear RNAs. Okay, so the nuclear RNAs are actually what are catalytic. They're actually going to do the enzymatic work here. But these are protein RNA complexes. And they bind to several places, okay? The first place they bind is at the 5' right here, the 5' exon to intron boundary. So they have one SNRP that binds here. They have another SNRP that binds at the 3' intron to exon boundary, so right over here. So they kind of flank the introns. Then there's a final SNRP that binds at an A near the end of the intron. Okay, so these are the three sites. At the first boundary between the exon and intron at the 5' region, the boundary at the 3' region, and then this A here. And this A is going to serve an important role. Okay, so these SNRPs join together, so they kind of join together to form these loops, right? And when they join together, we call them the spliceosome. Okay, so now we have a spliceosome that's assembled. And what happens then, so these come together, and this allows this A to attack the nucleotides over here at this boundary, this intron-exon boundary at the 5' end, okay? So this A attacks over here and breaks this bond, this phosphodiester bond, right here at this 5' intron to exon boundary. So the intron is cut right here from the exon, And this shape here forms a lariat. Okay, so this is another term for a lariat is a stem and loop. Okay, so you might have seen a lariat necklace, but it's just this stem and loop. So the final part, right, so we have this lariat formed. The final part is that the final intron-exon breaks. So the intron is released and the exons are linked together. So the lariat is cut out and the two introns are linked together. This intron ends up getting degraded and a phosphodiester bond joins those exons. So it's just one continuous piece of mRNA. So that's how splicing works, and we're going to talk about a little bit more about this in the next video. So the final parts of getting to a mature mRNA is the addition of this 5' cap. So there is a modified guanine nucleotide that's added to the 5' region of the mRNA. And this enables ribosomes, once in the cytoplasm, to bind to the mRNA. And it also protects the mRNA from nucleases that could degrade it. And so this is important to make sure that the mRNA is stable. The other part here is that here's this poly-A signal, right? So this poly-A signal is in this, we call this the untranslated region. This isn't actually part of what will make the protein. So this poly-A signal is what signals for termination. And it's also going to signal for this addition of a poly-A tail. So this is at the end, this isn't actually encoded in the DNA, but there's this long addition of 100 to 250 adenine nucleotides, and this is needed for translation. And this also protects the mRNA transcript from degradation. And it's added after cleaving the 3' end. Okay, so just to kind of summarize what we have here, we have our coding region with our introns removed and our exons fused together. We also have several parts that will not be translated. So we have the 5' untranslated region, or UTR, and the 3' untranslated region. And to each of these regions, we have our 5' cap and our 3' poly-A tail. Okay, so you should be able to know the parts of these mature mRNAs that are found in eukaryotic cells and what end they are at and what purpose they serve in the cell. So to summarize what you should know before coming to class, eukaryotic genes have several stretches of intervening non-coding DNA called introns, and the expressed portions of genes are called exons, and they're joined together by this process of mRNA splicing. mRNA splicing involves a complex of proteins and small RNAs. These are called SNRPs that form the spliceosome that cut out the introns. And as I'll talk about in the next video, many mRNAs can be spliced in more than one way. And then finally, this 5' cap and poly-A tail are added to the mature form of the transcript, and it's ready to leave the nucleus and be translated into a protein. So that's it for this video, and on to the next.